I think let me deal with this. Can you see my screen? Can you see my screen?
Yes, we can.
Yes,
we can see your screen.
Okay, good. Thank you. Well, thank you for the invitation, Professor Oyele. You know, when I got the chat, I can't reject that from my teacher, and therefore I have to accept. And when I was thinking, you know, it took me about 3 or 4 days to think of what do I want to present. And the reason why is because this is currently what I'm doing. And as Dr. Berry presented, this is one of our contributions to the body of knowledge, to abilities we were able to provide a low-cost opportunity on how to eliminate the lipofusion to fluorescence in the aging brain. And therefore, I think, okay, this one will not be useful so many because many people are not doing the human brain study in Nigeria, because we still have problem with the ability to get the postmortem brain, and therefore I did not present it. And again, while I was at LSU, this is one of our work that we published in 2020. I also think this one will not work because since some of the sophisticated procedure, we don't have them in Nigeria. Therefore, there's no reason for me to do that. Here, we use different in vivo electron recording, which is a very good protocol that if you have it at home, that would be a very good procedure for us to use. If I came back to this, that prenatal methamphetamine exposure on the developing fetus. And one of the reasons why I took the project was around late 2019. I saw this in the news that the unborn baby tested positive for methamphetamine. That was in Missouri, in US here. And when during the news, they discovered that the house where because the parents were surprised because they never used the drug. And they were trying to, during the course of investigation, they discovered that the house where they they purchased or probably rented, that it was once the factory for the methamphetamine production, the illegal production. And that's trigger, based knowing surely that my PhD actually focused on the MDMA and the nicotine co-administration. Therefore, that triggered me to use the drug. Fortunately, where I was here back in 2016, the drug was purchased for me when we were having problem with MDMA. I just went back to the freezer and I brought out the drug and we tested. And as you can see, Nigeria is having a pandemic now. I think we are having a pandemic. When my work started in 2012. There's less attention to there was a less attention to the drug of abuse, especially MDMA and methamphetamine in Nigeria. But before you know it, around February 2012, the NDLEA discovered a factory in Lagos where they manufacture it. And before you know it, they discovered another one in that was February, and in December the following year, they discovered another one again. And that made us to write this review, which was published in the journal, Nigerian Journal of Neuroscience. And the attention that I was trying to raise that time was that the risk of amphetamine-related stimulants is not only to the user, but the general populations. And I think this one of the copy of this work was sent to the NDLEA as well, because they are the one that gave me permission to use the drug. Being likely, I don't know, but it's likely I'll be the first person to use the MDMA as a drug for research in Nigeria that time, which I got approval from the NDLEA. And during that period, we projected in our review that since Viagon has been discovered in Lagos, that there's a likelihood there will be a factory around the eastern part of Nigeria, and likely the triangle, because in the drug cartel always want to, they use either triangle or they use a rectangle to coordinate their factory because they want to send within the country. Being Nigeria, being the Lagos being the source, we projected that they are likely to have one in the east, and probably they should, the NDLEA is also to focus their satellites in the northern part of Nigeria, especially the core north. And over the following years, we discovered that the drug was discovered in Anambra again, that they have a factory there. And before you know it, they caught a woman in Sokoto, as you can see, and that was late this year, which means seems to be that our prediction in 2015, 2016 actually seemed to come to pass. And the question is, why am I interested in the pregnancy, as I think I've told you earlier, because of the report from Missouri. And when I look at it, the reason why I choose methamphetamine is because it crosses brain-blood barrier. Though that's what they call it, because they call it crystal meth. They have one name in the East in Nigeria that they call it, and but it's not usually pure because it's combination of the derivative of it, but majorly they call it methamphetamine. But we got this, and the reason why, because it cross brain-blood barrier, it cross placenta blood barrier as well. And you can see because of the method of actions, the serotonin and the norepinephrine actually released, and they also cross the placenta barrier. And not only during the gestation, but during breastfeeding as well, they have their presence in the breast milk. And when we have 4 major negative consequences of the methamphetamine use, is first of all because they block the uptake of the dopamine in the system. Therefore, you have excess dopamine and the glutamate in the system, because once because they compete with that in transpositions of the dopamine, and that's very, very bad thing, because it results in the toxicity. And it has been reported also that the block, or as a result of that, it produces oxidative stress, and the oxidative stress actually have negative impact on the mitochondria, which is the powerhouse. And you know that as a result of that, once the methamphetamine binds to the receptors on the brain, on the neurons precisely, it produces the dopamine quinine, which actually trigger the inflammations, and it's sent from the resting microglia to the reactive one, and that also send the pro-inflammatory cytokines, which the disease molecule also secreted back, and that is a negative feedback that leads to the end results, which is inflammations, and the cascade continue. And as a result of that, the oxidative and the mitochondrial dysfunction also continue, and the glutamate toxicity will result in the product. And therefore, as a result of that, the aim of the work was to determine how prenatal and or postnatal exposure to the methamphetamine in mother affect the cognitive development and brain cognitive and the brain development. And what we did was we have 2 groups of saline, and we started with methamphetamine 4 milligram per kilogram weight, and we allowed it was administered subcutaneously, as we are through the fall of the back, because they can't use the peritoneum because of the gestation. And we administer all through the gestational period, which is very taxing, because determining the day 0 can be very stressful. And we continue, and after parturition, when they deliver, we separated this saline group into two. They call them the group SS group, and we look at the SM group as well, which means this group we continue to give them saline, and this SM we start now giving them the same dose that this group received, which is 4 milligram per kilogram body weight. While in the methamphetamine group, we divided them to two as well. We stopped some group, the group MS, we stopped giving them the drug, while the we giving them the drug, which means the SM continue to receive drug, continue to receive drug, while SS and MS stop receiving, the MS will stop receiving drug, we give them saline. If I will continue to use SS and MS and SM and as the case may be. And thereafter, we test for behavioral tests, the battery of tests, and we, after the test, after 60 days, we sacrifice the animal, and we process some for Western blot, some for fluorescence microscopy, and we did the some of the brain for the RNA sequencing. And while during the recording, because we did a lot of vocalizations and injections, actually we noticed this, as you can see, that between these two subgroup, we have the saline group less anxious compared to that disturbance. Without disturbance, they are actually very, very restless. You can see them, and that gave us, in fact, I did not even notice, it was I think one of the TA that time, the RA, the young student that would call my attention to it. I said, wow, if I noticed that, and we took record of that as well. And that led us to say, okay, because actually the original design was to check for the sequencing, because that was the intention. Hopefully that we'll do that later. And we check for the locomotion. We use the open field test, which is a classical test for locomotion activity and anxiety. And we discovered that the distance travel actually was impaired in the group that continue to use that we continue to administer with the MDMA. And when we compare with the group, was the group that we gave the methamphetamine during pregnancy, and we stopped, and you can see the significant difference between the two. And though the SM group had a lower distance travel compared to the control, there wasn't any significant difference. And therefore, we move further to check for the anxiety, as I've shown earlier. We discovered that the same group have a very low mobility time, which means their freezing time increases invariably, and that is a marker for anxietic behavior. And therefore, we say, okay, let's consider the cognitions. Therefore, we use a classical test, the Y-maze. I believe most of the people know this. The Y-maze, we put it here, and we allow the animal to explore. This is actually the battery test for special learning memory. This is mostly hippocampal dependent form of memory, and we discovered that there is no significant difference in the memory index. As you can see, it's the distraction rate is almost the same, and you can see the value here. Therefore, we do not see any single difference. Therefore, we went further to check the same form of spatial working memory, but this time around is a non-spatial one, which is a PFC dependent memory. And we saw it just as we saw with the anxiety and distance, the locomotion activity. There was a significant difference between the group that continue to use the MDMA. You can see the comparing the control to this other group that the saline before that were giving MDMA postnatally. There was a significant difference, and the same thing with this as well. And I believe most of people know how it's done. This is the trial one where we use 5 minutes. The intertrial interval is allowed to use between 40 to 40 between 30 to 50 to 60 minutes. But in this case, we use 45 minutes. And for the retest, we use the 5 minutes where you change one of the objects into the novel object. And to calculate the definition index, normally if the animal has learned, the animals will spend less time exploring the old object, while the animal will spend more time with the new object. But you can see that the MM group and the SM group, they could not discriminate much. And for our RNA sequence, the gene sequencing, this upregulated death score showed the black one shows those ones that are not significant that we look for. With the red one is the significant. Now let me explain. This bars is showing us the adjusted p-value, which means the significant one is down here. The lower p-value is here, while the uppermost p-value is up. And this dot is showing the sign, the number of genes that is there and the pathway that we are looking at. Therefore, you can see all these red ones are the one that are upregulated. The one that's around this one and that have a very significant value. And they can see that we have the AKT pathway is highly upregulated, and we have the MAC pathway also, so the long-term potentiation pathway, they are highly upregulated. Glutaminergic pathway, highly upregulated. We have cholinergic. I'm pointing to those pathways that are significant to drug of abuse, mostly. Then you can see the neurotropin signaling pathway, they are highly upregulated as well, though they have a fewer genes. And glutaminergic pathway, dopaminergic pathway, which is very consistent with what has already existed in nature, in the literature. The cholinergic pathway, they are highly upregulated. The SYNCAM pathway is very highly upregulated, and so on and so forth. Even at the axonal guardians, the reason is very important because the axonal guardian is very, very important in the wiring of the brain. Therefore, surprisingly, amphetamine addiction, the genes that are involved in amphetamine addictions are highly upregulated, definitely is expected. Then, and the same thing, this is when we are comparing MM versus SS, and this is MS versus SS, and this is SM versus SS. I can see that the pathway, though we have some of them are not consistent, but I think the one that have most upregulated genes is the MS, which is consistent with what we had with the behavioral test, with the other genes, the one that I've shown earlier. Okay, then we can see now this is the downregulated genes. Surprisingly, we saw these genes is very downregulated and it's very significant value, not significant value here. It is the same thing with the SMSS, SMSSS, and the MMSS here. You can see the nicotine addiction gene is highly downregulated, and the neuroactive ligand receptors also very upregulated. And therefore, we to analyze it again, because we want to isolate those genes that are majorly affected, that we can process them further, since we cannot analyze all the about 2,000 genes that we have in the analysis. Therefore, we did the volcanic plot. The volcanic plot, looking at the volcanic plot, this is log or 2 or 4 change, and why this one is p-value, the log of p-value. The line here we see is the p-value. Anything above it is what is significant. Anything below this p-value line is not significant, as you can see. And this on the my left side is the one that are downregulated, while on the right side are the upregulated genes that you can see. And you can see here, when you look at the SM versus SS, you can see about 628 genes downregulated, while the 360 are upregulated, and so forth. And the same thing with if you compare. But if you look at the MS as just as I showed you earlier, you can have a very significant number of 888 genes that are upregulated when you compare the MM versus SS, which is the same thing that you can using this pie chart to show. And we then try to pull it further because we can't process all of them. Then we wanted to say, okay, let's use this pathway to see which one is mostly affected. And so to our surprise, we see CAMK2 alpha very upregulated. You can see this in red. You check it. You can see green 2, some other green 2, CAMK2 beta, green 3 alpha, and AKT3, ADCY1, and IGFR1 is upregulated, which is very good. It was a good news for us, and that makes us to check them to confirm that in our using our Western blot. Therefore, we confirm the phosphorylated CAMK2 level, and we discovered there was a elevations of in MM, and especially on the SM. Those groups that are continually giving the drug, they are highly elevated. And surprisingly, this time around also, between the drugs, the MS group also have a very significant lower one compared to the rest, but no significant difference when we compare, which means the impact, it seems to be waning off after we stop the administration, which means the reinforcement is very, very important. Continuous administration of the drug is important to produce the effect. And then we check for the PCAMK3 as well, AK3, and we saw that unlike the PCAMK2, the SM and MS no significance when compared to the control, while the MS, inconsistent with what we saw in the behavioral study, was highly significant. And we check for the ADC. This time around, I can show you the SM data. I think the antibody didn't work well, but we'll see, trying to see what we want to do about that. And there was a very significant difference when you compare the SM to SS to the other two groups. Now, what does that mean in summary? Now, if you remember what I showed you earlier, that the ADC is very, very important. Why? Because when you look at the literature, the methamphetamine bound to it. And ADC is a very important enzyme in catalyzing ATP into molecules, and which is calcium dependent. Therefore, if you increase the ADC in the sample, definitely you are highly likely to increase the influx of calcium, which is not good. It is not a good thing for the cell, because if you over increase your calcium in the system, definitely you are likely to overload your cell, and that is very bad for polarization and repolarization. And the same with the AKT. AKT, the three pathways, they are very, very important for transcription factor in the nucleus, and they are all upregulated. But on the AKT3, it was downregulated, which have affected. Therefore, the next thing we wanted to do is to, okay, let's go and check the health of our cells. But as I said earlier on, we have the anxiety in the sample, we have the locomotion problem, and we have the spatial learning deficit, which is especially the PFC dependent form of memory. the next thing we want to do now is to characterize the major cells of the nervous system, which is the microglia, the astrocyte, the oligodendrocyte, and the neuron, to see how healthy are they. But because of my time, I think it is the best time for me to stop. And let me acknowledge the people. This is my friend, Dr. Bunele Amita. This is Dr. Tolu. This is my Dr. Lee, my PI, who gave me the postdoc offer before I moved down here. And this is Tolu, is the one that took over from me. It's like this is the sent forth party for me that time. And thank you. This is our campus here at OHSU. You are welcome to come and visit it. And I hope you mark your calendar for this. Don't forget this. Thank you. And tomorrow I will still be presenting. And on Fridays, Dr. Ajayi will be presenting on our AYS platform. If you're a young person, you are welcome to join us tomorrow and Saturday. Thank you for your time. I hope I did not overshoot time.
Thank you. Thank you, Dr. Philip. You finished 5 minutes earlier. Yes, thank you for keeping to time. So, like I have typed on the comments section, if you have any question or comments for discussion, you can feel free to type it on the chat box, or you can raise your hands to be given audience to speak. So. Okay. There's a question here. Let me read it out. Thank you very much, Dr. Adeniyi, for this wonderful work. I need to clarify on the time at which the administration of both saline and MA were terminated in the progeny. Also, what informed the choice of dosing of the MA, that's the 4 milligram per kg body weight?
Okay, thank you for the question. I think I said we only stopped the administration when the mother delivered. Therefore, we give the animal, and I think I forgot to mention that we are not giving every day. We are giving 2 days apart. The animal we are giving, they are injected twice in a week, kind of. Therefore, we stopped only when the mother delivered. For those that we want to stop, for the rest, for the other one, we continue administering to the mothers. Don't forget, this is the mother that we give, not the offspring. Until the mother, until they stopped taking the drug now stop when we win, which happened to be 27th day after delivery, because we in the animal house we stopped. You win the animal after 27 days, and you know after that time they can't breastfeed again. Then there was involved the choice of 4 milligram. Actually, it was my PhD, because during my PhD, the drug I used 2 milligram. I just choose to double the dosage this time around, since the MDMA is more toxic to the dopaminergic system compared to the methamphetamine. That's the reason why I increase the dosage twice of what I use for my PhD work. I think that answers your questions.
Okay, thank you. Yeah, thanks for that response. We have some questions coming up. Another question, someone wants to know, someone has been trying Onwe Obinna, he has been trying to assess this drug and have been having difficulty, especially with amphetamine. Do you have any suggestion you can give him?
Yes, I think the best way is, I believe, if you are a student, what I did that time was I wrote the NDLA, and the NDLA give me the drug. Normally, you cannot use the drug. You can't use, because these drugs are controlled drug. MDMA that I use is, I think, the Schedule 1, which means it should not be given to people as a gift, and it must not be sold. Therefore, you can only get them through the NDLA in Nigeria. You can't even purchase it from outside. In fact, even if it is wrong, you can't even publish it even if you get it on the. Therefore, the best option is for you to write the NDLA. NDLA, tell them what you want to do. You can probably, if you want to know, I can check my letter that I used that time. Therefore, but what you need to do is just write them, calculate the dosage you want to do. Don't forget, just make sure to me, I try to increase it, like probably I added miscellaneous to it to make sure that I won't have to write them again. That's what I did, and they send it to me. When they send it, they send me with their seal. And when I'm traveling, I go with the old seal, that's in case if I meet them on the way. I think that's how. But if you want to use methamphetamine, the one I use for this work, you can actually buy the methamphetamine chloride. It's available. It's available in like a syringe like this. You can buy it, but I'm not sure if the NDLA will allow it to enter Nigeria. But in US here, that was how I was able to beat it. You can buy the methamphetamine chloride. Yes, it's available in market, but I'm not sure but the best way is for you to approach especially if it's for your postgraduate study, approach the NDLA, and it is for free. They will send it to you. Right? I wrote to the chairman of the NDLA and tell them what you want to use it for, and let them. You have to account for every degree. In fact, that's the reason. When I was talking, I talk about that. I sent the review to the NDLA because it's like when we publish every paper, I send it to them, and I wrote to let them know how much I've used. I report to them about once a year. Thank you.
Okay, thank you for that response. Yes. So another one, someone wants to know, were there any consideration on whether the drug interfered with pregnancy development?
Yeah, we did not see, because none of them, we did not have, there is no, except probably one or two of the animals that were not able to take in. Probably we believe that probably the animal failed to take in before we started the drug, which means it seems to affect, I think, probably one or two female. We saw that, but we did not take that, since I did not focus more on the reproduction. I'm more interested in the one that succeeded in pregnancy. But we did not see any abortions or abortions or something, miscarriage, none. And there is no teratogenic effect.
The next question. Okay, thanks for the response. Another question goes like this. Is MA capable of affecting cognition if administered before pregnancy?
Before pregnancy? Well, we did not consider that. Well, anyway, that should not affect it. If I saw that the Professor Oremosu is here, neuroembryologist. Therefore, I'm not sure, and even for our basic embryology, we know that I'm not sure it will affect the egg in the mother, and that may not likely to affect. Therefore, the effect is likely to be during because of we administer all through either from the first trimester to the last trimester, from the onset. Therefore, that's the effect we are looking at. I'm not sure we'll see any significant effect. And that the reason why I focus on the interest of pregnancy is because this drug is usually used as a drug for raping and the nightclub and for among the young people, which some of them, they won't know when they are pregnant. They are likely to continue to use of the drug. And that was the main basics for why I use the drug in pregnancy.
Okay. Thank you, Dr. Philip, for answering the questions. It's already time for the second presenter to start his presentation. So we'd like to ask Dr. Philip, please, there are still questions here. If you could type the response on the chat box, maybe during the presentation, so that those asked could access it. We don't have time to discuss these questions now. So I would like to.
I saw my prof, Professor Emerson raised hands up. Probably.
Oh, okay. I didn't see
that. Yeah. So please, let's bring Professor Emerson, the senior person.
Okay.
Excuse me. Excuse me. Good morning, everybody. I want to first of all appreciate the presenter, Dr. Philip, for acceding. And I want to thank Professor Woyele, Professor Lokpade, all the senior people. It's just a comment to say that it's also important that we continue to work across subspecialties and not work in silos, and to appeal to Dr. Philip to also help to co-supervise anyone who is doing any work, in his related field. I know there are a lot of advances in the US that we are yet to catch up with. So I think we will just continue to leverage on the fact that some of us have been able to get into that system. So we congratulate you, and I won't want to waste time. Thank you very much. And I also appreciate all those who are on the platform. Thank you for allowing me to listen to this beautiful presentation.
Thank you,
I remember that I read through the entire work when you had to do your defense.
Yes.
So very much.
Well, it was you that gave me my doctorate. Therefore,
okay. We can see. Okay. But there's somebody else, please. Let's listen to somebody else raising up the hand, Carl Remington. Can you go ahead and talk?
Hello, have I joined the wrong meeting?
We are off to Neurotalk, NSA meeting.
I don't understand.
Okay. All right. Do you know him, Dr. Philip?
No.
Okay.
Yeah, I'm already removing. Okay, thank you.
Okay. So, Dr. Moses, I think you have the floor now for your presentation. You can share your slides. Dr. Moses, are you there?
Is my screen showing?
No, sir.
Yeah, it's started showing now.
Yes.
Okay.
You can put it in
play
mode
so that we can see.
Dr. Ekong, I think you can keep talking while you are trying to fix that.
Okay. Thank you very much. Thank you, Dr. Bere, for the wonderful citation, and thank you, the Neuroscience Society of Nigeria, for giving me the opportunity to share the information concerning publishing our data, especially the young ones who are yet still having problems with publications. My talk is about publishing data in a journal, and I'm giving the editorial viewpoint. Moses Ekong from the Department of Anatomy, University of Uyo, Nigeria. First of all, I will need to define some terms. Which will allow us to flow very well. So starting with who is an author. I decided to use a different approach to defining who an author is as an originator of a data report. This data report could be in different forms. It could be from experimental work, it could be from a field data collection. And the mere fact that it is able to be put into an article format, that becomes a report, which an author is the originator. Who is an editor? Editor is someone that ensures that the data reported from the author are well ordered. It is written in a standard form. It is organized in such a way that it makes sense, and is also arranged to suit the journal's criteria. Then the third person is who is a reviewer. And I define a reviewer as someone who inspects the author's data report. Someone that inspects the author's data report. And with that in mind, we now go into the crux of the matter. What constitutes an article? Or what constitutes a data report? Principally, an article is a report of a data by the author, which we have seen from the author's perspective. Report of data by the author, which is supposed to be written in a formal, concise, and unambiguous terms, while at the same time being guided by the fact that since it is a reported speech, it must be written in such a way that it reflects that. And at the same time, it should be written in a subjective form, which means that it allows someone to look at it critically, not as a personal item, but as something that every other person can look at as a realistic document. And most articles basically are organized in such a form as the title page, the abstract section, the introduction section, the methodology section, a result, discussion, and conclusion. As in the case of the neuroscience, the Nigerian Journal of Neuroscience, the result is separate, the discussion is separate, while the conclusion is also separate. But in some other journals, these can be as one section. Then the references. In some other journals, before the references, there is the appendix where you have other information, which we will see as we continue. So now, what constitutes a title page? A title page is usually made up of the topic, that is the title of the article. In the Nigerian Journal of Neuroscience, we encourage a running title to be included, which will be at the end or following immediately after the main title. Then the author's names could be as many names as possible, and the affiliations. Then the corresponding author's information follows. These are typical of what is seen in the title page. But in some journals, you can have it including the word count, the number of figures or tables, as well as even adding note to the editor as part of the title page. And as well as in other instances, you talk about, which we will get back in the course of the presentation. The abstract section, what is required in the abstract section? The abstract section must have a background, which may not be more than 2 sentences. And then from the background, we go to the aim methodology, results presentation, and the conclusion that is drawn from it. And these is usually within a limited number of words. The Nigerian Journal of Neuroscience usually adopts 250 words as maximum. And this also is what PubMed adopts. And the problem usually arises if you have more words. There's a tendency for PubMed to cut it off immediately. It is 250 words. So we always employ authors to limit the number of words to what each journal wants. But for ours, it's 250 words. Then the introduction section, what is expected from an author? The background of the study. It gives literature of what is happening, current literature, as well as that in not distant past. Then, is there a justification for this study? This must be incorporated as part of the introduction. And most times the introduction will end with an aim of the study, which allows someone reading the work to understand what the work is all about. The materials and methods, usually a detailed description of the different materials and methods utilized in the course of the work. And all of them usually comes with their own citations where applicable. The results, usually a step-by-step presentation of what resulted in the course of the methodology. You can have different steps in the method. So as far as there are results following, these results should also be replicated in the results section, which guides someone reading it. And in the process, tables are generated, figures are generated. So whichever is applicable, one needs to cite them while presenting the results. In the discussion and conclusion section, there is a step-by-step assessment and interpretation of the presented results. It assesses what was the result, what does this result imply, and what are these findings going to make. You then therefore go ahead to compare it with similar results, whether they are in line or they are in contrast. And if they're in contrast, you give an explanation why such should happen. That brings up a good discussion. And in conclusion, you provide a short summary of the findings and the inference or implications of such findings. These are the basic things one is expected to see in an article. Then looking at going ahead, where they have the appendix, which the Nigerian Journal of Neuroscience also accepts. You can put funding information. Did you receive a fund from multinationals or from funding agencies, or there was none? You just use your personal funds. Then you acknowledge those that help you are not authors, but help you in the course of the research. Was there any conflict of interest in this work that you did? This should be stated. And who did what? What did you do? What did author A do? What did author B do? These are the information that should be presented in the author's contribution. And if there is any other information that is important, it can also be included. Then the references, which should be formatted to suit the journal style. The Nigerian Journal of Neuroscience uses the modified APA system, which is easy to quote. So what is then constitute the editorial process? By the time an author finishes all that we've just finished discussing, he is to send the work now. He now sends the work to the journal, which in this case, the Nigerian Journal of Neuroscience. What does it entail? The Nigerian Journal of Neuroscience therefore receives the manuscript and it undertakes what is called editorial processing. And what does editorial processing involve? Or let me just look at the general processing. Generally, you have the editorial processing, a peer review, editorial decision is taken. If it is accepted, the article is published after payment of the article processing charge if necessary. If it is not, the article is rejected and a Gallup proof is formed and it is published. So looking at what does the editor do when he receives the article? The author submits the work to the editor through the, since the Nigerian Journal of Science uses the e-copy, e-based mode of article submission, he receives the article and then decides to process the article by checking what is the manuscript actually relevant? Is it a neuroscience base? If that, there is a check. Is the manuscript properly formatted in line with the journal's guide? If that is in order, there is a check. Is it the work original? Because there will be a check. Is this work available somewhere else? If it is original, there's a check then. Then the methods of generating data, are they reliable? Then there's a check. Do all the methods meet ethical standards? Then there's a check. If none of these, is the data enough to draw a valid conclusion? Then if any of these shows that is a no, the manuscript is returned back to the authors for revision to incorporate relevant information. But if item 1 is the problem, the manuscript is outrightly rejected. But if it is formatting, it can be revised. If it is not an original work, it's also rejected. If the methods of generating the data are not reliable, it is rejected as well. So these are some of the things that are involved before the journal now undergoes a peer review. So what is the peer review process? It is just to send to experts who can vouch. Like I said, an expert that inspects the work of the author. Is this author's report in line with standards? Are the methodology what is currently in use? Are the appropriate citations taken care of? So that is what the editor does by sending it to the reviewers to do all these checks. And since the journal operates a double-blind peer review process, the author does not know the reviewer, neither does the reviewer know the author. It's only the editor that knows both. And so this tries to limit bias from either side. So the author does not know the reviewer who is reviewing the article. So we try as much as possible to ensure that that is what it is. Second, also the reviewer does not know the author. So when the work now gets to the reviewer, what does he consider? He considers the work, whether it is original. And does it generally align with the title? Does it follow? Because at times some title could misrepresent what the article is actually about, or it could also over-exaggerate what the article is all about. So it looks at the work and looks at the title. Do they align? Is there an aim? And is this aim relevant and reproducible? Is there a novelty? Are the conclusions valid? This the reviewers considers and looks at also if the manuscript is well written. Can it communicate exactly what the author have in mind? Because at times while writing, you might think you have included the relevant information, whereas the information is within your mind alone and it is not reflected in the paper. So the reviewer looks at it. Does the work tells us exactly what the editor wants, the author wants us to know? Are there grammatical and typographical errors? If any of these is a no, the work is, the reviewer recommends for a revision, depending on what the level of problems he has seen. It could be a major or a minor revision. But if all is checked correct, it is recommended for possible publication and is sent back to the editor. And is sent back to the editor for consideration. So by the time the editor receives the work, he now cross-check what the reviewer did. Was everything done in a professional manner? And if it is yes, I don't know, my system seems not to respond. If the work is a yes, then it is the author is contacted and the manuscript is accepted. But if there are issues with the work, the author is asked to do revisions depending on whether it is minor or major. But if the recommendation is negative, the work may be rejected if 2 or more reviewers report the same as negative. But if it's a single reviewer that gives a negative report, it can be considered by the editorial team to determine if a revision is what called for. And this can be communicated to the author. I don't know, I don't seem to be able to move my slides any longer.
You can stop it and start share it again.
Can you reshare?
Okay.
Well, while you are trying to do this, can you continue to talk?
Yes. Like I was saying, if the work does not meet up to the standard of the journal, through the recommendation of the editor and the reviewer, the editorial team considers it and takes the decision whether to reject or to call for a revision, or to ask for the authors to reconsider their work in another journal. So if it is a pass, the work is published. The work is published. If it is for revision, the work now is sent back for revision and the process continues again. That is, the work then comes back to the editor and he now sends it back to the reviewers for their input again. If it is okay now, it becomes a pass and the work is published. But if it comes back still negative, it is rejected outrightly. and the author may consider submitting to another journal or going to the lab again to do more or to add more data to make the work actually relevant. So, in conclusion, if authors follow the editorial guide, the chances of publishing the data report in journal is much higher. And so I use this opportunity to invite us to the special issue of the journal. For those who did presentation at the Ibadan conference, you still have a deadline of November 15 to send in your article for publication in the special issue of the Nigerian Journal of Neuroscience. But for the regular issue, we are still also collecting paper, and we are calling for volume 14 for 2023 for publication of the regular issue of the Nigerian Journal of Neuroscience. So I acknowledge the Neuroscience Society of Nigeria for giving me this opportunity to present this information. And also my predecessor, Professor Amadi Hongu, who guided me as his managing editor to the point that I am able to take over the responsibility of managing the journal. Thank you for joining the webinar.
Thank you, Dr. Moses, for this wonderful presentation. So, and thank you for keeping to time. You stopped exactly in 30 minutes. So it's now time for question and answers. And okay. Yes. Can you please talk about publishing a review which doesn't have materials and method? That's one question.
Well, a review, okay. Publishing a review is based on, it still has the materials and method section, although it is not really highlighted that much, because there are methodology which involve in searching for the literature. What are the sources of literature you are getting the information from? How relevant are they to the work? And how many were sourced? So this forms part of the methodology for a review article. And also there are some review articles that has to do with getting it is the same format. It's just that the methodology is much less compared to that of an experimental work.
Okay, thank you. I think we have a hand up. Let me see.
Professor Way, can go on.
Good morning.
Professor Way,
Yes. I want to thank Dr. Ekong for this wonderful presentation. I want him to talk very briefly about what constitutes authorship. You know, he told us the originator of a data report. But for those who are just coming, some are wondering, is it only the person that conceived the idea of the research, or those that practically participate to generate data, or those that write the first manuscript, or those that read the manuscript, and those that give the final approval to the manuscript? I want him to talk briefly about what are the other components of what actually makes authorship. Thank you very much, Dr. Ekong.
Thank you very much, Professor Woye. Well, authorship, there is no strict sense of authorship, but there are criteria that determines who is an author. First of all, yes, there's someone that needs to generate the idea. And there are people that sit down to discuss this idea to bring it into something that makes sense. They are qualified to be authors. Then the people that now gather data, they are qualified to be authors. And the people that do critical review of the work, whether at the supervision level or as pay. They are also co-authors. They are also part of the authorship of an article. But authorship does not involve people that fund. If you give money for a research, it doesn't make you an author, except you have a direct scientific input into the article. Thank you.
Thank you for that response. We have another hand up. You have the floor.
Dr. Faborodi.
Dr. Oluwaseun.
Okay. Good morning, everyone. Thank you, sir, for the presentation. I have a number of questions I would like to have some information about. The first one is regarding references. I want to know if Nigerian, the journal has a reference style that can be found in Mendeley, EndNote, and all, so that it might be easy for people to easily format their references automatically. And then I also want to ask about the article publishing charge, if there's a charge, and if there are opportunities for waivers or discount, depending on whatever the journal decides. And I also want to know about the indexing options for the journal, which indexing opportunities are available for the journal. And I also want to ask about the plagiarism level. What is the accepted plagiarism level for the journal? And also, if journal has a chance for checking for double submission, in case where someone submits a paper to the journal and has also submitted the same paper to another journal, just to look for which one comes out first, if there are ways by which the journal would check for double submission. Thank you very much, sir.
Well, thank you very much. The comments are much, starting with the article processing charge. The journal takes a charge of 30,000 naira only for article published in the journal, and that article will appear online as colored and on printed form as black and white. But if one wants the articles to appear as colored in printed form, it will have to pay an extra 10,000 naira charge to it. Then as for if there are discounts, for now, we are not having any discount because we don't have sponsorship. So, but if we get the sponsorship, which we intend to apply for, there will be opportunity for us to have discount and even free publication from time to time, as the case is. But actually, there is a discount for the special issue, which was because they presented at the neuroscience conference. So that discount is there. Then on plagiarism check, yes, we do have a plagiarism. We check for plagiarism and we keep the level at 15%. Anything that is more than that, we don't outrightly reject a paper, but we ask the authors to revise their work to meet up to the standard. Because while the journal is there to publish high quality work, it is not our best to reject our papers, except where we see where it is irredeemable. So we encourage people to publish, but at the same time, if you don't meet up to the expectation that we want from you, we are forced to reject your paper. As for checking if there are other publications, yeah, we do not have that facility right now to check if there's double submission. But actually, we believe that authors should be ethically conscious to know that it is unethical to submit papers, one paper to two different journals at the same time. Then as for indexing, we have each article published has a unique digital object identifier, which is in cross-ref. So we are indexed there. Then we are also indexed in WorldCat OCL. Google Scholar indexes us, as well as site factor. However, we are under evaluation for the Web of Science indexing, just as that of PubMed Central, which we believe will come out successful. Since it's usually a long process, it is something that we applied since earlier in the year, but it's still undergoing different, because the evaluation is a different level. So we are still at that level till now in those ones. Thank you. I believe I answered all your questions.
Yeah, this is Tohir Fayaz from India. My question is that there is a concretizations and abstract. So in what way the abstract has to be concretized? And there are lots of journals are there. That is original article which is mentioned, that is original article, original research articles. Too many disparities there. So in what way, how we can concretize the abstract? In what way? So can you explain about that?
Okay, if I get you very well, Prof, you are asking if we have a particular way in which we want our abstract to come up. Is that what you mean?
Yeah, that's my question.
Okay.
That is abstract. That is also in philosophical terms that we want to know abstract to concretize. That has to be in the publications. So that the thing which we are not knowing about this. So in what way abstract to concretizations? That this is that is the way. And so many journals which I've seen there in the neuroscience article, that original research article, that is in the original articles. So these are the disparity among this research journals. So on the basis of that, they impart that there is an impact factor of this. So how
Okay.
we can judge it? Yeah, how we can judge it? The abstract which we are not knowing about this, and concretize, we have to concretize it. Concretizations in terms of that is the word work, the original works. So in some journal they say original work. So is there any disparity among this? That's why I'm asking about if you have a, yes, you can say better in the way.
Okay. We do not have any disparity between whether it's a original research work or a review or a case study. All the abstracts follow the same format, which is a single paragraph of not more than 250 words, which must have information on the introduction, the background, the aim, the methodology, results, and conclusion.
Now these all are the mentioned. That is the protocol. Yeah, that's a protocol. Yeah. This is the protocol for this one. But in Nature, Lancet, New England Journal of Medicine, and then some Indian journals, Indian Academy of Neurosciences. So article is going to be published there. So the written thing is that. So my question is that is an abstract to concretize what way we reach to this. That is a journey. That is the ideology of the human mind. If you know better,
Well,
then you can explain that.
well, I don't,
Left time.
I don't, Professor Fayez, I don't really understand your question again.
Okay. No problem.
It's okay. Thank you very much.
Okay, thank you, Dr. Moses. There are lots and lots of questions lined up for you. But we've taken up the time for this webinar. So I would like to ask the president, do we accommodate more time to answer the questions, or would he respond to them?
I think we just maybe just spend more like 3 or 4 minutes briefly for him to answer the question.
Okay. Okay, so I'll go with the next question. Someone is asking, what are the likely conflicts of interest in publishing a paper? What are the implications of conflict of interest?
Okay, one likely conflict of interest is if a pharmaceutical company gives you money to do an experiment in their drugs, with their drugs, and possibly the experimental results were not favorable, they will not want you to publish it. So that is the conflict already in it. And also, if a government agency gives you money to carry out a research and the report does not seem to favor the government, they may not want you to publish it. So for you to go ahead to publish, you must declare there's a conflict.
Okay. Then, yeah, I think the next question, you've already answered it. What's the publication fee? Okay, so the next one is, please, sir, the author reviewer double blind, what constitutes or informs the choice of reviewers?
It is area of research.
How do you select your reviewers?
Yeah, it is based on the area of research. We have a lot of reviewers amongst us who are experts in different fields. So whatever field of interest of research that is an article comes, we look at the list of the reviewers who is in this area, which can give an informed decision concerning whether the article, the methodology is right. That is what informs sending an article to a particular reviewer.
Okay, thank you for that response. Another question, can a paper be rejected or sent back for review because of old references in the manuscript?
Well, when once an article has been received and we note some information, we write back to the authors asking them to effect those decisions. So we expect the authors to do that. But if at the end of the day the authors refuse to, the article can be rejected.
Okay, so quickly, the next, to just answer this quickly, the corresponding author, what qualifies corresponding author among the authors? That's one of the questions.
That is depending on the authorship. It could be the most senior person in the lab, or it could be the first author. It could be the person who initiated the idea. Everything, it depends on them. It's not the journal that determines that.
Okay. What is the impact factor of NJN, Neuroscience Journal of Nigeria?
Well, like I said, we are under evaluation. So those impact factor will be out before the year runs out.
Okay. So someone is asking about originality of a research work. Is it wrong to repeat a work that has been done in a different lab?
Well, if a work has been done in a different lab, has the work been published? If it is published, then you cannot send it out to an article to be published again, except what you are trying to say that the work that was not done properly. So that is not ethically allowed because it is someone's work already done already. So why would you want to repeat it again and publish?
Okay. So another question, please, what can one do in a situation where a paper is reviewed by the reviewer and later sent back to the author? The author revised and sent back as it was given to another reviewer. And a part of his review contradicts the first reviewer. Okay, he's trying to find out what if reviewers' comments contradicts one another.
That is left for the editorial team to handle. The editorial team will handle that. That is not the business of the author. The editorial team will handle that.
Okay. Someone is asking, for suggestion, kindly address the issue of an author withdrawal of his manuscript and plagiarism.
Well, if after an author sends in a manuscript to the journal, and for whatever reasons, at whatever stage, and they think the manuscript can no longer be published in the journal, they can write directly for withdrawal and it will be withdrawn. But if the article is already published, that would take a longer process and a lot of other communication because it has already been shared to the world. So that is that. But if it has not been published, yes, it can easily be withdrawn. As for plagiarism, like I said, we always check and advise the authors on what to do and what not to do to make the work unique.
Thank you for that. The next question, throw more light on first, second, third, and last authorship.
It is not the role of the journal to determine who is first or not. That is left with the researchers and the authors to arrange themselves, because it depending on the climate. If you're talking about the US, the European nation, you're talking about Europe, you're talking about the supervisor is usually the last author, while the student who carry out the work is usually the first author, with the postdoc being the second or third author, depending on how many people worked on it. Whereas in the Nigerian situation, it is not like that. So it is not, that is why we wouldn't dwell in who becomes first author or not.
Thank you for that response. The last question is, must the research and results be carried out in Nigeria? Sorry. Yeah, must it be carried out in Nigeria only, or can it be from other country?
As far as the lab is an endowed lab, we have the necessary facilities. Even if it is done in mass and brought to Nigeria, we accept it. We are trying to be even though it's a Nigerian journal, we are trying to be an international journal. So we are not a local journal. Thank you.
Okay, thank you so much for the responses and for a wonderful presentation. Thank you for the comments so far from Professor Remusu and Professor Fayaz from India and everyone here. So we've come to the end of this webinar. I don't know if the president has a word to say before we close.
Yes, let me thank you, our uncle, the secretary, our able secretary, and for the presenters. I don't know, we have a Professor Lokpade here who graciously canceled the lab meeting for tomorrow to pull in the audience, and he helped to spread this, advertise this webinar. I don't know if Professor Lokpade will want to say one or two things.
Thank you very much, Mr. President. Nothing to add but to appreciate our speakers. It's great to see Philip moving up and up and up. I think after we met 10, 12 years ago, when he was, and Moses and the way he dispatched questions was that he's not been a figurehead editor all these years. He's been hands-on. So we are proud of that. We are seeing that there's progress. I guess the only challenge I will give to our speakers is to continue the hard work and train other people. So 5, 10 years time, you should also be listening and people presenting work that is educating you, people talking about the journal in a way that you never thought it could be like that. It's more higher. And then as a society, in neuroscience society in Nigeria, we know we're tremendous. And to the president and the executives, I mean, to pull, there was a time the attendance was almost close to 100. I think we've come of age. We should just keep doing things well, ethically, international best standards. And I'm very proud that neuroscience in Nigeria will reach for the heights. Thank you very much, sir.
Thank you, Professor Lokwadi. On that note, I want to appreciate our presenter, Dr. Philip Adeniyi, for obliging to give the presentation at very short notice. As he has said, I just told him, and I knew it shocked him, and I told him I want a research-based presentation. I know he does all these, bringing up the youth, and I really appreciate the presentation. Thank you so much. I want to also say that it's about 2 a.m. or 3 a.m. where he is, so he has really sacrificed to be up and to be able to. And also technically, he has assisted us in hosting this meeting today. I also want to appreciate our editor. I also want to appreciate our editor-in-chief for the enlightenment. I think just as has been said, everything was concise. And I use this opportunity to invite us to submit to the journal. That's the only way we can have something that we are really proud of. I see that the level of patronage is not too good, but I just realized that it is not that if we are not writing manuscripts in Nigeria, because by virtue of have been on the editorial board of another journal, I know we have quite a lot of submissions. So please, those that are neuro-based, please let's try to send in some of them to the Nigerian Journal of Neuroscience so that we can actually develop it and it to be something that we can all be proud of. So that's the basis of bringing our editor-in-chief, so that if you have these questions to ask him, you can ask him, and you can always continue the talk even after the presentation. I want to thank the members of my executive and the society, the members of society, and the participants at large for turning up in this large number. And I want to sincerely apologize that we had to use double link. It is to make the presentation very smooth. Thank you so much for attending. Certainly we'll continue this tradition. It's not going to be a one-off. So I want to appreciate every one of us. Over to you, Dr. Ebere.
Okay, thank you, Mr. President. I would like to announce that we've come to a close of this webinar, and you'll be notified before time when we'll be having the next webinar. And we want to also encourage you to show us your support and participate as you turn out in this webinar. Thank you so much, and have a great day for those in Nigeria, and have a good night's sleep for those in America. Thank you.
Thank you.
Dr.
Thank you.
Thank you, Mr. President.
Thank you.
And thank you, everybody.
Mr. President. Thank you,
Presentations.
everyone, for participating.
Thank you, Dr. Ekong. Dr. Moses Ekong. Well done.
Nice presentation.
Dr.
Nice
Abraham Musa, nice to see you.
Dr. Abraham,
Dr. Uwe, thank you.
how are
Thank
you?
you.
Dr. Philip.
God bless you.
Thank you, sir.
Dr. Philip, God bless you.